Hello everyone, welcome to Get My Class. So let's solve this question here. It says identify A, B, C, D, E and F. And they have given the structure here and they have given A, B, C, D, E, F. Okay. And the options they have mentioned A as antigen binding site, B as antigen binding site, C as disulphide bond, D as light chain, E as light chain and F as heavy chain. Option B, A as antigen binding site, B as variable region, C as disulphide bonds, D as heavy chain, and E as heavy chain, and F as light chain. Option C says A as antigen binding site, B as antigen binding site, C as sulfide linkage, then D as heavy chain, and E as heavy chain, F as light chain. Option D says A as antigen binding site, B as variable region, C as sulfide bond, D as light chain, E as light chain, and F as heavy chain. So let us see. Now, if we see this structure, what is this? This is nothing but antibody. What is this? Antibody. We all know antibody is a Y-shaped structure, right? It is a Y-shaped structure. Okay, now when this is, what is this? This is antibody. Now, antibody is made up of two chains, okay? Like two types of chains, in fact. So this is called as the light chain. What is this? This is the light chain. It has two light chains. Okay, two light chain. This and this also is a light chain. Okay, so E is also light chain. Light chain. Okay, and here and it has a heavy chain. So these long ones are heavy chain. So here F, which is marked here, it is a heavy chain. Heavy chain. Now, here in the light chain and heavy chain, in the beginning, this is the region where antigen binds. So this is antigen, you can say antigen binding site. Binding site. Okay, which is also called as a variable site or variable region because it will change itself depending upon the type of antigen. So variable region. So B is variable region. Here again, this is also variable region. So A is also variable region, or you can also say antigen binding site. Binding site or variable region, both are same. Now here, these chains are bound with each other by disulphide bond. By disulphide bond. We know that usually in proteins, you know, the structure is formed, the folding of the chain is formed by the formation of disulphide bonds, right? So here you can find the C which is marked, that is a disulphide bond. Here this is also disulphide bond. Here also you can see disulphide bond. Now this part, this is the variable region I said, right? And this part is called as the constant region. Okay, constant region of light chain. This is the constant region of light chain. Light chain. Okay, so that is a light chain. So D can be, you can consider as a light chain itself. Okay, so let us see. Option A says A is antigen binding site. That is correct. B is also antigen binding site. C is disulphide bond. D is light chain. Yes. E is light chain. Where is E? Yes, that is also light chain. Then F is heavy chain. So this is F, heavy chain. That is absolutely right. Okay, so option A is correct. Now option B says A is antigen binding site. That is correct. B is variable region. That is also correct. Option C is disulphide bond. Correct. D is heavy chain, which is incorrect. So we can rule it out here. Then C says A is antigen binding site. Correct. B is antigen binding site. Correct. C is sulfide linkage. It's not sulfide, it is disulfide. So it is ruled out. Option D says A is antigen binding site. B is variable. Correct. C is sulfide bonds. No, it is disulfide bond. Okay. So that's how we can just mark that option A is the correct answer. Let us conclude. Which is the correct answer? The answer is option A. I hope you understood the explanation. Thank you, everyone.